Hello, everybody, and welcome to our Innovative Workflows in Drug Discovery webinar. We're so glad that you're here with us today. Please grab a snack or beverage, sit back and relax. We will begin shortly when all participants have joined. Okay, great. So then let's get started. Welcome, everybody, to the webinar again. We're talking about innovative workflows in drug discovery today. And in particular, we're focusing on drug interactions and dosing with the I.DOT. Thank you for being here today. My name is Holger, and I'm the head of product management here at Dispendix. We have registrants from all over the world, and I'm so proud to be moderating this webinar today. On the agenda, we will hear from an exciting panel of experts in drug interactions and drug dosing. Please feel free to use the Q&A feature through our program. We will address your questions at the end. Let's get started. Before we bring our esteemed speakers to the stage, I'd like to take a moment to introduce the groundbreaking technology that's at the heart of the discussion today, the I.DOT. For those who are unfamiliar, the I.DOT is an automation-friendly, non-contact dispenser that revolutionizes the way we handle liquids in the lab. Let me highlight some of its key features. The I.DOT can transfer volumes as small as 2.5 nanoliters up to 30 microliters. It's compatible with 96, 384, and 1536 well plates, offering incredible flexibility. It can handle a wide range of liquids, from methanol up to 65% glycerol. So now let's dive into how it works by watching this short explainer video.
The I.DOT is an automation-friendly, non-contact dispenser designed to transfer nanoliter and microliter scale volumes to 96, 384, and 1536 well plates. 96 well I.DOT source plates can dispense any liquid with viscosity from methanol to 65% glycerol. One channel of the dispense head and one well of the source plate combine to form a dispensing channel. The dispense head modulates air pressure, which can be precisely adjusted to generate droplets from 3 to 50 nanoliters at a nanoliter resolution of 0.1. Each channel generates 100 droplets per second into the target well. A 96 well plate can be filled with 100 nanoliters per well in 7 seconds. To conserve valuable reagents and samples, the I.DOT requires less than 1 microliter of dead volume. To provide users with the highest degree of process safety, the I.DOT counts the number of droplets dispensed with miniaturized light barriers mounted underneath the source plate. If source wells run out of liquid during dispensing, the I.DOT can backfill the missed droplets, preventing user error. The I.DOT enables fast, precise, and traceable liquid handling, enabling the most efficient and reproducible genomics, drug discovery, and synthetic biology workflows.
Okay, great. So with this introduction, I hope everybody now has a basic understanding of the I.DOT. And this technology will be central to the innovative workflows our speakers will be discussing today. And now, with no further ado, let's welcome our distinguished speakers to the stage. The first speaker is Dr. Basil Wicki, who will be presenting the landscape and molecular underpinnings of mycobacterial drug interactions. Basil is a postdoctoral researcher at the Department of Biomedicine Basel, focusing on bacterial mechanisms of treatment failure, particularly in Mycobacterium abscessus. With a microbiology background from the University of Basel, he has gained extensive research experience at Bioversus, the Swiss Tropical and Public Health Institute, and during his PhD at the Department of Biomedicine Basel. His research has centered on investigating resistance mechanisms in Mycobacterium tuberculosis and Mycobacterium abscessus. With a passionate commitment to addressing antimicrobial resistance and developing innovative treatment strategies for challenging pathogens. Basil, please take it away.
Thank you, Holger, for this very nice introduction. I'm very happy to be here, and let's see whether the control works. Maybe I have to click the slide again. Here we go. Perfect. I'm very happy to talk about drug interactions today, particularly in Mycobacterium abscessus. And let me give you a very quick introduction to this bacterial pathogen. See, here we go. M. abscessus is an emerging pathogen which is highly abundant throughout the environment. It can be found in lots of different sources such as soil, water, and household devices. M. abscessus is especially dangerous for people with a pre-existing pulmonary disease, and it can lead to very severe lung infections in these people. Okay, here we go. So in recent years, we saw a drastic increase both in incidence and prevalence worldwide for M. abscessus, making it an important pathogen to study in more detail. And the main importance really comes from the fact that M. abscessus is highly multi-drug resistant, drastically narrowing our options we have in the clinic to treat an infection. When we look at the typical treatment regimens against an M. abscessus infection, we always have to use a combination of multiple antibiotics of different classes, which are administered over a very long period of time. But when we look at the outcome after 6 months, upon giving this very drastic and strong treatment approach, we see that in 2 out of 3 patients, M. abscessus is still present in the sputum, meaning that in 2 out of 3 treatment approaches, the treatment actually ends in a treatment failure. And we think that this combination approach obviously is the correct way to treat M. abscessus, but we also think that we so far understand too little about underlying mechanisms leading to this treatment failure we see currently in the clinics. And one of these underlying mechanisms we think could be drug interactions, which can occur between drugs when they are administered simultaneously to a patient. And to study these mechanisms in more details, we decided to systematically assess drug interactions in Mycobacterium abscessus. In order to do so, I first had to establish an experimental pipeline. And I really wanted to focus on a simple pipeline, which allowed me to go on a high throughput approach, but at the same time also be very precise and accurate. Let me show you quickly how this pipeline looks like. So first, I used automated system, particularly the I.DOT, to dispense compounds to a 384 well plate. I then used 96 channel pipettes and again the I.DOT for certain approaches to add the bacteria to these plates. These plates were then incubated, and after 3 days, optical density readout was conducted. And with these OD readouts, I then analyzed the data using an in-house analysis pipeline. So as you can see, this is not a complicated setup, but what it allowed me to do is to assess more than 120,000 unique drug combinations and measure more than 1.5 million growth assessments. At the start of this project, there was a growth inhibition screen of a diverse library of around 2,600 compounds. This library consists of many different compounds such as antibiotics, human targeting drugs, but also food additives and many different other molecules. And by screening this library based on growth inhibition, I identified 48 compounds which are defined as being active against M. abscessus. And as you can see in green, most of them are antibacterial compounds, but also some other classes of compounds were present there. Then, as a first step of combination, I took these 48 active compounds and combined them with each other in a two-pair drug combination approach. And what you now see here are so-called checkerboard assays. And in a checkerboard assay, two drugs are increased in their concentrations along the X and the Y axis, and the growth of the combination is then assessed, what you see here in black and white. This entire experiment you see on the right side consists of more than 1,100 checkerboards, around 500,000 growth assessments, used almost 1,384 well plates, and was about 1 month of work. But what I would like to highlight is that all this effort only took 6 I.DOT source plates. So by using efficient setups of work lists and the I.DOT, it is possible to go from a very simple I.DOT source plate and dispense very complicated patterns of drug combinations in my case. And this can be done in minutes per plate, allowing me for a throughput of up to 384 well plates per day. When we now look into the results of the drug interactions, this is the picture we get. And when I now zoom in to one of these checkerboards, in this case clarithromycin and clofazimine, I can quickly explain to you how I assessed drug interactions. Based on the inhibitory concentration of 50% of the single drugs on the left and at the bottom, I could use a drug interaction model that predicts along this purple curve how much concentration is needed to inhibit 50% of growth if the combination is not interacting. And by actually measuring these concentrations, these IC50 concentrations of the combinations, what I've done with the green dots, I could assess whether I have an antagonistic, a synergistic, or no interactions. And in this case, as you can see, as the green dots are on top of the purple line, I don't see any interaction. However, back to the matrix on the right side, we see that reality is quite different. Most of the measured combinations indeed show a drug interaction, and of them, again, 60% roughly show an antagonistic drug interaction, meaning that, as in this example between azithromycin and amikacin, the concentration necessary in the combination of these two drugs is higher to achieve the same effect than what we would achieve by just using one or the other drug by itself. And it is important to mention that many of these antagonistic combinations I assessed are actually used in the clinics nowadays to treat M. abscessus, which could be an indication, to some extent at least, why the treatment failures we currently see, why we see these treatment failures currently in the clinics. The good news is I also identified very strong synergistic interactions. And one example I would like to highlight is this combination of the rifampicin and imipenem, so a rifamycin antibiotic and a beta-lactam antibiotic. And this combination is, for several reasons, not yet used in the clinics against M. abscessus, but it highlights the potential of combinations with a very strong synergistic effect to be studied in more detail and hopefully find better combinations and treatment regimens in the future to treat pathogens such as Mycobacterium abscessus. From this, basically a small data set of 1,100 combinations, I wanted to increase the number of combinations. And what I did is I took my 48 active compounds on the y-axis and combined them with all the inactive compounds of the drug library along the x-axis. And what you see on this plot is, despite one of the two drugs being completely inactive against the bacteria, we still see a considerable amount of drug interaction. And with this amount of data, we could ask the question whether we could identify chemical patterns that can describe the mechanisms of drug interaction. In order to answer that question, I took these 2,500 inactive compounds, here each dot representing one of these compounds, and clustered them in a so-called tree map based on their chemical similarity. So the closer two of these dots are to each other, the more chemically similar these compounds are to each other. And I could then add the drug interaction data with one active compound against these 2,500 inactive compounds as a color map on top of it. So in this example of tedizolide, an active compound, and the 2,500 inactive compounds, I was able to identify clusters of drug interactions, so showing strong synergy, such as the beta-lactams or a bunch of psychiatric drugs, or a very strong antagonistic cluster of the fluoroquinolones. So this approach indicates that by measuring systematically a high amount of combinations, we can potentially find chemical signatures that describe drug interactions, which then can help in drug discovery and drug development in the future. So far, I've only talked about drugs and their influence on drug interaction, but the question arose whether also the bacteria and the genetic background could play a role in this whole story. And to answer that question, I took 8 clinically relevant antibiotics and combined them in every possible two-way combination, so 28 combinations in total. And I then assessed the drug interactions across a panel of 269 mycobacterium clinical isolates, each isolate of a unique patient and having a unique genetic background. And what we see on this picture very nicely is that the drug interactions are highly heterogeneous across these clinical isolates, indicating that indeed the genetic background seems to play a role when it comes to drug interactions. Another way to assess this question is to measure the so-called heritability. With heritability, we can define how much of a phenotypic variation, in this case, the drug interaction variation, can be explained by the genetic background of the bacteria. And as you can see on the right side, for some of these tested combinations, more than 50% of the drug interaction phenotype can be explained by genetics. And since all of these clinical isolates were whole genome sequenced, we could run a genome-wide association study approach, here with the example of amikacin and clarithromycin, and we identified genetic variants which are highly associated to the drug interaction phenotype. This is an ongoing effort, and we are currently working with knockouts, complement mutants, and so on to answer in more detail what exact genetic mechanisms underlie drug interaction. But for now, I would like to finish and summarize the project as follows. We first showed that drug interactions are very common among clinically used antibiotics. And we think that the high amount of antagonism, especially in currently used combinations, could to some extent explain the high treatment failure. We characterized chemical signatures of highly interactive compounds, and we think that approaches like that will heavily influence, to the positive, I hope, the future drug discovery and drug development efforts. And we revealed that not only drugs are important when it comes to drug interactions, but that also the genetic background of bacteria is a driver of drug interactions, implying that in the future it is very important to understand the bacteria in more detail to optimize treatment approaches against these pathogens. And it is an ongoing effort to identify the mechanism of drug interactions on the bacterial genetic background. Overall, this study highlights that with a simple approach, but very strong and efficient mechanisms and methods, we were able to assess a wide variety of combinations and go into details of patterns of drug interactions. And I hope that this effort will support the future finding of more efficient and better treatment regimens against pathogens such as Mycobacterium abscessus. And with that, I'm at the end of my presentation, and I would like to thank the entire Boeck Lab, especially my supervisor, Dr. Lukas Boeck, who guided me throughout my entire PhD and where I'm still here for my postdoc. Every team member who participated, everyone did to some extent, and all the collaborators and all the funding agencies. Thank you very much.
Right. Thank you so much, Basil. That's really impressive data. And don't forget to type your questions into the Q&A section so that Basil can take these questions afterwards. Great. So thank you again, Basil. Great data. So our next panelist is Francesca Mastroianni, who will be presenting Streamlining in Vitro Toxicology, Precision Drug Dosing with Non-Contact Liquid Handling Technology. Here we are. And Francesca earned her bachelor's degree in biotechnology from the University of Basilicata, followed by a master's degree in functional genomics from the University of Trieste. She completed her master's project at the University of Helsinki Institute of Technology, HighLife, and her research explored the role of microRNAs in regulating neuroprotective factors and protein aggregation in Parkinson's disease. Currently, Francesca is a laboratory scientist at UCB Pharma based in Braine, Belgium. She's been contributing to cutting-edge research at UCB since 2021, and she has extensive experience in cytotoxicity and neurotoxicity assays and advanced imaging technologies. Her expertise includes neuroid outgrowth real-time imaging assays, assessment of neural network activity using microelectrode array systems, and various proteomic and genomic technologies. As the lead of the cytotoxicity assay within the investigative toxicology group, Francesca is instrumental in assessing compound safety during the early stages of evaluation. She oversees the execution of cytotoxicity assays, which act as a first filter to assess the toxicity properties of all compounds of interest, identifying any potential toxic effects before compounds can advance further in the development pipeline. So, Francesca, the stage is yours.
Hello. Yes, thank you, Holger. I'm just requesting control if you... Okay, perfect.
There you go.
Thank you. Okay, so good afternoon, everyone, and thank you for joining this webinar today. So I am going to share with you our advancements in streamlining in vitro toxicology through precision drug dosing by using non-contact liquid handling technology. So today I will walk you through the journey that brought the I.DOT into our laboratory and how it has changed our workflow. So let's move on to the agenda for today. Okay, so we will start with some background, then we will explore how I.DOT is being applied into our investigative toxicology lab. We will then talk about the future prospect of I.DOT uses and applications, and finally we will wrap up with some conclusion. So let's start with some backgrounds. So first of all, in our experimental workflows, we faced several challenges before introducing automation. One of the main issues was the limited throughput, because the number of compounds we could test per week was restricted due to the time-consuming nature of manual processes. Additionally, we often encountered reproducibility issues, as the variability in manual processes can often lead to inconsistent results, which could compromise, of course, the reliability of the data. Manual intervention requirements were another significant concern, as manual handling is known to be error-prone, increasing the risk of mistakes. We were also concerned about the... It's not working. Sorry. Okay. So we were also concerned about the operator-dependent variability, which could result in changes in the outcome of the experiment based on who was performing the task. And lastly, our previous setup had limited flexibility, making it difficult to adapt the workflow to different experimental conditions. So to address all these challenges, we needed to select the optimal instrument based on several criteria. First of all, the budget we had available to purchase a new equipment, the ease of use and training of this new equipment, and its adaptability to future demands of the lab, but also the ability of the new equipment to speed up operation in the lab, and of course, the ability to generate trustworthy results. So our journey to integrate the I.DOT into our investigative toxicology lab involved several stages. We began by identifying equipment that aligned with our demands and capabilities. This involved assessing various options and, of course, determining which technology would best meet our needs. Then we conducted demonstrations to evaluate the performance of different instruments, and after thorough assessment, we selected the I.DOT as the most suitable equipment for implementation in our lab. So at this point, we prepared all the necessary budget requests for the purchase of the I.DOT, and we obtained the necessary approvals. Following this, we began with the execution of the validation processes to ensure the reproducibility and the confidence in the assay after the integration of the new technology. Of course, we also provided training session and support to all laboratory staff to make sure that everyone was able to fully use the I.DOT with high standards and high efficiency. And finally, we fully integrated the I.DOT in our experimental workflow, enhancing our capabilities in the lab. Now, let's talk about the application of I.DOT in our investigative toxicology lab. One of the most important assays in our lab is the cytotoxicity assay, which is a cell-based assay used for screening compounds to determine if the test molecule shows cytotoxic effect. Now, this assay plays a critical role in the progression and selection of molecules, enhancing the confidence in using these compounds for downstream testing. So the assay begins with the seeding of cells, followed by the compound dosing, which is the step where the I.DOT has significantly improved our workflow and productivity. Now, after the incubation, we perform the ATP-based viability assay to measure the effects of the compounds on the cells and generate a concentration response curve to determine an IC50. At this point, the entire process concludes with the generation of a report and the publication of the data into the database. So the integration of I.DOT has brought numerous benefits to our lab. First of all, it allows for rapid and precise dispensing of compounds, allowing us to bypass the need for a serial dilution step, which is prone to human errors and inconsistencies, since this step often leads to inaccurate concentration levels and compounds binding to plasticware. Also, these automated dispensing systems has allowed us to significantly reduce the manual intervention and minimize cross-contamination risk. By automating the dispensing process, I.DOT minimizes variability, ensuring greater reproducibility and uniformity of results. It also offers high flexibility for allowing for customization of experiments to meet all specific requirements and experimental conditions. And lastly, this technology also enables us to test a larger number of compounds in a shorter amount of time, helping us to increase the turnaround time, the throughput, and also save time. So by mitigating all these aspects, the integration of I.DOT has significantly improved our assay reliability and efficiency. Indeed, after the integration of I.DOT in our workflow, we have observed substantial time saving in compound testing, allowing us to test more compounds than we could ever do before. Indeed, this improvement has streamlined our workflow, and we are now able to achieve more in less time. So as you can see from the graph on the left-hand side of the slide, the amount of time needed to test around 50 compounds showed a significant reduction when we compare the manual dosing and the I.DOT dosing. So, of course, this allows us to handle a larger number of compounds in less time, reducing the overall turnaround time of our assay. Also, the increase in the number of compounds tested can be observed on the graph on the right-hand side of the slide. So, as you can see, there is a significant difference between in the number of compounds tested after the I.DOT integration. Because basically this technology enables us to produce data with a faster turnaround time, with higher quality, and this also helped us increasing the confidence of our stakeholders in this assay. So looking ahead, we aim to further enhance our capabilities in the lab. So first, we plan to expand the use of I.DOT to additional assays, streamlining workflows and further increasing productivity and capacity. We would also like to evaluate the possibility of combining I.DOT with other robotic systems to generate a fully automated or semi-automated workflow, further reducing manual intervention and, of course, maximizing efficiency and reproducibility. And additionally, we also aim to develop miniaturized versions of existing assays to reduce reagent consumption and cost, while maintaining high quality of the data we generate and high throughput. So, in conclusions, by automating key aspects of our experimental workflow, we have achieved several critical improvements. First of all, the I.DOT technology has proven to be an efficient and valuable tool for drug dosing in cell-based assays. By focusing on automation, we have improved our experimental capacity and quality while reducing the overall turnaround time of our assay. We conducted extensive validation process to ensure a reliable and robust assay, also providing consistent and dependable results. Of course, by reducing reagent consumption and minimizing the need for repeated experiments due to variability, we have achieved cost savings. And finally, the transition to an automated dispensing system has significantly reduced manual labor, freeing up valuable time for us to focus on more strategic work. So overall, the adoption of I.DOT technology represents a significant leap forward to streamline in vitro toxicology testing by enhancing research capabilities, improving the quality of data we generate, and ultimately contributing to the development of safer and more effective drugs. So with this, I thank you for your attention, and I am happy to answer any questions you might have or provide further information. Thank you.
Thank you so much, Francesca. That was really impressive. Thank you so much for outlining your experiences in toxicology with the I.DOT. And yeah, now it's time for our Q&A session. So let's jump in and answer any questions that you might have. And we do have a couple of questions in the chat. But please feel free also to give additional questions in the chat or in the Q&A section. We still have sufficient time to answer them. So the first question I see here from Gerald is to you, Francesca. And the question is, hi, Francesca, what made you decide for the I.DOT compared to other instruments that you assessed for your workflow? So what were the factors? Why did you go for the I.DOT?
Yeah, so Of course, one of the key aspect that we had to consider was the budget that we had available. But also, like I would say that the main aspect that made us choose the I.DOT was the ease of use of this instrument, because it's quite simple to set up the like a protocol and integrate the I.DOT into our workflow. So that's for sure the easiness of use of this instrument. And then it also for us also the dimension of the equipment was also important, because since we use the I.DOT for dispensing on cells, we need to maintain sterility. So these the I.DOT we were able to put the I.DOT inside a cabinet, so inside a hood, to keep the sterility. So I would say that, yeah, it was a great choice for us.
Great. Thank you so much. And then we do have another question to you, Francesca, relating to cells. So Lisa is asking, hi, Francesca, did you try seeding cells with the I.DOT?
No, we didn't, because we have another pipetting robot available in the lab. So for the seeding, we are using that, but it's something that we might explore in the future. It might also be useful to use just one instrument to seed the cells and then do the treatment.
Great. Small comment from the dispensing side of things. So yes, the I.DOT can seed cells. There are plates with different pore sizes. We've got a 200 micrometer pore size that is suited to seed cells, magnetic beads, antibodies, proteins. With cells, obviously the density is important, right? So you can only seed cells with a certain density. So that's something to keep in mind, that obviously they also need to be separated well, not to clog the pore. But yes, the I.DOT can also be used for seeding cells.
If I may, just as a small comment, we did the whole last experiment with the clinical isolates. This was all done with the I.DOT, also the bacterial dispensing. I know bacteria and cells are not per se the same, but just saying, if you have a lot of different cells, this worked very good for us, very reproducible. Obviously, the sedimentation of the bacteria is probably the main problem to clogging, so it has to be homogenized, and you need to be a little bit faster than with compounds generally.
Yeah, great insights. Thank you so much. And then, Francesca, I would also have a question more technically related. So I was wondering, in the workflow you presented, were there any particular challenges, and do you have any particular wishes for, equipment, lab equipment in general, or towards the I.DOT? So do you have a wish list to make your workflow even better?
Yeah, so I guess that when you want to integrate a new equipment into your workflow, you always need to make several adjustments to ensure seamless operation. So of course we had to make several adjustments to make sure that the I.DOT was fitting into our workflow. So we modify our protocol. So we had to adjust the volume of cells that we had in each well. We adjusted the concentrations, how we were doing the dilution. So when we were doing the assay manually, we did the whole serial dilution manually in a separate plate. Now with the I.DOT, of course, we always need to consider that we cannot go over a certain amount of the MSO. So we get the compounds at a specific concentration. So we are not able to dispense all the concentration of our concentration response curve, starting from a single this stock concentration. So we had to find the best range of intermediate concentration to make sure that the I.DOT was able to to dispense at its best. And then, of course, you also need to adapt your SOP and all the, since this is a routine assay, we have a very strict procedure. So we had to optimize all the SOP and make sure that all the people working in the lab were completely training on the use of I.DOT and were fully independent on how to handle problems or like solving problems and so on. And then about the wishes, I would say that probably my wish would be to have a platform where we could somehow integrate the I.DOT with other robotic system, especially automated plate handling systems. So in this way, basically this setup will take on the burden of microplate management. So the handling, the storage, retrieval and transport of the microplates, and it will of course significantly reduce manual labor. So it will enhance efficiency and also boost the scalability of the assay.
Mm-hmm. Interesting. So you're thinking about a robotic arm exchanging source and target well, so that you don't have to do this manually.
Yeah.
I think we're able to help you with that. Let's discuss it later.
Yeah.
Great. Okay. Thank you so much, Francesca. And now, Basil, we also have some questions for you. The first one is from Lisa, and it's again about dispensing cells. So Lisa is asking, hey, Basil, was there anything you did to make sure your bacteria was homogenized before dispensing on the I.DOT? Cell setting is an issue in our hands. We use HEC293 cells.
Yeah, it's a very good question. I mean, especially mycobacteria are heavily prone to clump. So we always work in these assays so far with single cell suspension. So we do filtration steps up front. I think this is for sure helping heavily with the clogging of the I.DOT source pores. Otherwise, homogenizing is possible, but then when they settle, then they settle. So it's all about speed. And I think the assays with all the different clinical isolates, it was a lot of manual changing of source plates, going back and forth and back and forth, a lot of refilling. So honestly, I'm not sure how this could be solved. Maybe have source plates on a shaker and then be very efficient with I.DOT. But for us, it was really a lot of refilling, next step in I.DOT, refilling, next step in I.DOT. Yeah, but I would say everything under maybe 2 minutes worked very fine. We maybe had 2 or 3 clogging issues throughout these 269 clinical isolates, so that's basically nothing.
Okay. Great. Okay, awesome, Basil. Then thank you so much for this answer. I have an additional question to you. So, I mean, resistance towards antibiotics and treatments of bacteria is a big topic, right? And so I think your research is really on the spot, and it's really like very helpful in general. So I was wondering what, from your perspective, would be the next steps, or are there any plans to translate your research into actionable items so that the public can benefit from it?
Yeah, that's always the question, right? I guess how do you translate that now to real life, to clinics? I mean, to be very clear about that, it's a high throughput approach. So it has a lot of limitations, and we also ignore a lot of factors. There's no pharmacokinetic and nothing like that. It's an in plastic in vitro assay. That said, I would argue 2 things. The first is it's basically MICs you measure, the change in MICs, and this is something that is used in the clinics every day, right? So the change in MIC, you could argue, seems to have, to some extent at least, some truth when it comes to clinical decision making. And by showing that this can change based on drug interaction, this might have some input. The other thing is, and this is what we are working at the moment, is you have to obviously then go to in vivo, or at least in vivo-like environments. And we are working on lung infection models, organoids, or air-liquid interface models, something like that. But then at one point, it also has to go, I think, to animals and then so on and so forth. But this is always the point with high throughput. You start very, very big, and you couldn't do that with in vivo models. And then you find interesting leads, or not leads, right? But interesting findings, and then you optimize and go further downstream.
Yeah, great. Thank you so much. And then the final question for you, Basil, same as for Francesca, any particular challenges, any wishes, any developments you would love to see in the future from a technical perspective?
Yeah, it's interesting. I mean, we have different other assays in the lab where we are working with agarose. I mean, this is not now my project, but there it is all about temperature control, for example. I'm aware that different I.DOT systems have temperature control currently. If I'm not mistaken, I think one of the newer I.DOT has it. So that's certainly something that we are looking into, to have a temperature-controlled I.DOT, also to dispense at 37 degrees with agarose gels and so on and so forth. And if I go very far, I talked about in vivo-like systems. I would think it would be fantastic down the road to work with high-throughput organoid systems. And who knows, maybe the I.DOT could be used when we can seed cells. Maybe it could be used to dispense organoids. I'm just putting it out there. I don't know, but if we have at one point 384 well plates full of organoids and testing all kinds of compounds and combinations in high throughput approaches, yeah, for me this sounds quite amazing, to be honest. So this direction.
Okay, exciting. Yeah, cool. I think with that we should be good for today, unless there are any other questions, or if you have anything else, please put it in the chat. If not, if any questions come up later, we're also more than happy to answer them via email. So feel free, if after the call you've got anything you would want to talk about, reach out to us directly. And with that, I would like to thank you all for attending the webinar today. And no matter where you are in the world, we thoroughly enjoy connecting with the scientific community. And of course, would love to hear your feedback. So there will be a short survey and a webinar recording included in a follow-up email. So stay tuned for that. You will receive an email shortly. And yeah, with that, all I can say is have a great rest of your day, and looking forward to connecting with you the next time. Bye-bye.
Bye-bye. Thank you.
Thank you. Bye.
Bye-bye.